Hello guys, this is Tanveer. In this video, I'm going to discuss glycolysis with you. But before glycolysis, I would like to discuss some of the basics. So let's move to the basics. As you know, whatever we eat, maximum parts are carbohydrates. Actually, what happens that in the GIT, that carbohydrates will get break down into the smaller forms. That means the polysaccharides will break down into monosaccharides like glucose, galactose, fructose, etc. So in this video, I'm going to discuss the metabolism of glucose. So let's see what will happen with glucose. As you know, in the jejunum of a small intestine, the glucose will get absorbed. You can see here, glucose is here, and glucose will get absorbed by a transporter that is called as SGLT1. That means glucose will get absorbed in the enterocyte from the intestinal lumen. As you can see, the name SGLT1, that means sodium glucose transporter 1. That means if glucose will get transported inside the enterocyte, with that sodium will also get absorbed, right? That means sodium will come inside. As you can see, with each molecule of glucose, sodium will come inside the cell. That means there will be so much sodium inside the cell. So there should be some way to remove the sodium out from the cell. So here you can see here sodium potassium ATPase. This will do this function, that is, this will move the sodium out. This will move the sodium out, and with that potassium will come inside, right? And this is the sodium potassium ATPase, that this will use energy in the form of ATP. SGLT1 is not using energy, but because of that, sodium potassium ATPase is functioning. That means indirectly SGLT1 is using ATP. That means this process is called as secondary active transport. This is secondary active transport, right? Now glucose will come inside the enterocyte, and from this enterocyte glucose will move out. So glucose will come here in the intercellular space, and from here it will move in the plasma, right? My question to you is that where does the SGLT2 present? Please write down in the comment box. Okay, so glucose will come in the plasma, and from the plasma this glucose will move into the cell, right? It will move into the cell, and this function is done by GLUT transporters. And let's assume that the cell which I'm talking about, this cell is a cell of skeletal muscle. That means GLUT4 is present there. I would love if you will write down in the comment box that what are the other locations where GLUT4 is present, right? So glucose will come inside the cell. In the cytoplasm of the skeletal muscle, glucose will come and it will change into pyruvate, right? Pyruvate. It will change into pyruvate. This process, the conversion of glucose to pyruvate, this is called glycolysis, right? This is called glycolysis. This is the topic of discussion today. That means conversion of glucose to pyruvate, this is called glycolysis, and we will discuss this today. So what will happen next? This pyruvate will move into the mitochondria, and the transporter here is pyruvate proton symporter. That means with pyruvate, proton will come. Okay, and what will happen with this pyruvate? This pyruvate can change into acetyl CoA. This pyruvate can change into acetyl CoA. This pyruvate can change into oxaloacetate also. And with this there will be a cycle that is known as Krebs cycle, and this Krebs cycle will produce NADH, FADH2, etc. Right? These NADH and FADH2 will go into this chain of reactions that is called electron transport chain, and in the end of it, oxygen will accept the electron and it will change into water, right? And the proton which will come in this lumen, right, these protons will move through this F0 F1. This will change ADP to ATP. So that means finally we will get ATP. In the end of this whole pathway, we will get ATP from ADP. You can see we have discussed many things here. There are many questions arises. The first question is, what is the location of glycolysis, right? What is the location of Krebs cycle? What is the location of link reaction? What is the location of electron transport chain? You can answer all these questions from this discussion. You can answer the transporters. One more thing I would like to discuss here only, that all the enzymes of glycolysis are present in cytoplasm of cell. Cytoplasm of the cell. All the enzymes of glycolysis are present in the cytoplasm of the cell. So this is the overview, and now let's move to the glycolysis in detail. Right. Okay. As I told you just now, that glucose will enter inside the cell through a transporter, that is GLUT4, right? So the glucose will enter. Here you can see the glucose will enter inside the cell. And the characteristic of this transporter GLUT4 is that glucose can enter through the transporter and can exit as well. So as soon the glucose will enter inside the cell, we have to do something so that it will not be able to move out. For that purpose, we need to add something on the glucose, and we will add phosphate, and glucose will change into glucose 6-phosphate, right? Glucose will change into glucose 6-phosphate. The question arises here is that from where we will get this phosphate? The answer is that we will get this phosphate from ATP, that means adenosine triphosphate. This ATP will give us a phosphate and it will change itself into ADP. And as we extract the phosphate from ATP, and we will add that phosphate on glucose, and glucose will turn into glucose 6-phosphate, and it will not be able to move out from the cell now. That means it has been trapped inside, right? And one more thing is that, what is the enzyme here? The enzyme here is hexokinase. Hexokinase. At some places there is glucokinase as well. In the next video we'll discuss the difference between the two. For now, just remember that hexokinase will be used here. As you can see in the name, there is kinase. That means if you'll see the kinase, just think that there's something about the ATP, right? That means we are using ATP here. Okay. So what will happen next? Glucose 6-phosphate will isomerize and it will change into fructose 6-phosphate. Fructose 6-phosphate. Fructose 6-phosphate. And what is the enzyme here? Of course, isomerase. So write the name of the enzyme here, isomerase. Right. The next step is very important, and why it's very important? Because one more phosphate will get added, fructose 1,6-bisphosphate. And who will give this phosphate? Again, ATP will give this phosphate, and we'll get phosphate from ATP, and ATP will change itself into ADP. The name of the enzyme. The name of the enzyme here is phosphofructokinase. Very famous name. Phosphofructokinase 1. Phosphofructokinase 1. Again, you can see kinase. Why this step is so important? This step is so important because this is the rate limiting step of the glycolysis. If this reaction has occurred, that means glycolysis will occur. All right. Fructose 1,6 bisphosphate. I would like to say one more thing, that there are three reactions in this pathway, glycolysis, which is irreversible under the physiological conditions. In some other circumstances, these reactions can be reversed, but in normal physiological conditions, these processes are irreversible. The first one is this, which is catalyzed by hexokinase, this one, and the second one is phosphofructokinase 1, right? And the third we will discuss in the second part. As you can see here, we are using ATP, right? That means this is the first half of this glycolysis, in which we are using energy in the form of ATP, but in the second half we will get energy in the form of NADH and ATP. So we are spending money, and in the second part we will earn, right? Okay, let's move further. What will happen this fructose 1,6 bisphosphate? This fructose 1,6 bisphosphate, in the presence of an enzyme which is known as aldolase A. Aldolase A. In the presence of aldolase A, fructose 1,6 bisphosphate will break down into dihydroxyacetone phosphate. Dihydroxyacetone phosphate, right? Dihydroxyacetone phosphate. And the second molecule here is glyceraldehyde 3-phosphate. The second molecule here is glyceraldehyde 3-phosphate, right? Glyceraldehyde 3-phosphate. So and you can see here, they are acetone, right, and aldehyde. That means they are isomers of each other, so they can convert into one another. So after the breakdown of fructose 1,6 bisphosphate by aldolase A, we will get dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, right? This was the first half, and what will happen next? So let's move to the next part. As you can see, dihydroxyacetone phosphate can change into glyceraldehyde 3-phosphate. So let's assume that now we have two molecules of glyceraldehyde 3-phosphate, two molecules, and so on. Every reaction will occur twice, right? In this step, glyceraldehyde 3-phosphate will get a phosphate in inorganic form. In which form? It will get in inorganic form. A phosphate will get added in inorganic. That means ATP is not giving phosphate here, right? We are getting a phosphate that is inorganic, and it will add on the glyceraldehyde 3-phosphate. And the one very important thing which will occur here is that we will get NADH from NAD plus. And how many molecules? Of course, 2. I told you everything is 2 here, right? That means we will get 2 NADH here, and we are adding a phosphate in inorganic form. This molecule is 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate. Bisphosphoglycerate. 1,3-bisphosphoglycerate, right? 1,3-bisphosphoglycerate. And let's write the name of the enzyme here. What should be the name of the enzyme? Of course, glyceraldehyde 3-phosphate, as we are removing the hydrogen in this reaction. So in the name there will be dehydrogenase. So glyceraldehyde 3-phosphate dehydrogenase, I will write in the short I will write in the short, that is glyceraldehyde, Ga3P, glyceraldehyde 3-phosphate dehydrogenase. Dehydrogenase, right? Glyceraldehyde 3-phosphate dehydrogenase. That means we are getting 1,3-bisphosphoglycerate. And the next point is, what will happen with this 1,3-bisphosphoglycerate? Now it's time to get energy in the form of ATP. So this 1,3-bisphosphoglycerate will release a phosphate, and that phosphate will get received by the ADP, and that will change into ATP. And of course, how many? Of course, 2, right? So we are getting 2 molecules of ATP here. That means phosphate will get removed. So the name of the next molecule should be 3-phosphoglycerate. 3-phosphoglycerate. And what should be the name of the enzyme here? The name is phosphoglycerate kinase. The name of the enzyme here is phosphoglycerate kinase. Phosphoglycerate kinase. Right. So we will get phosphoglycerate kinase, and we will get 2 ATP here. And it will shift from 3rd position to 2nd position. That means it will change into 2-phosphoglycerate. 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 And the name of the enzyme here is phosphoglyceromutase. The name of the enzyme here is phosphoglyceromutase. 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 Right? We'll get 2-phosphoglycerate. Right? And what is the next step? In the next step, this 2-phosphoglycerate will change into phosphoenol pyruvate. Phosphoenol pyruvate. Phosphoenol pyruvate. And the name of the enzyme here is enolase. It's important name. You will get questions from here. Name of the enzyme here is enolase, right? And 1 molecule of water will get, of course, if it's 2, then it's 2. Molecule of water will get removed here. Water will get removed. And the next step is very important, in which we will get, again, we will get energy in the form of ATP. So ATP will change into ADP, will change into ATP. Of course, it's 2 molecule, right? Pyruvate kinase, right? Pyruvate kinase. And the final product, that means pyruvate, or you can also call as pyruvic acid, pyruvate. So we will get pyruvate, that is the final product here. So as you can see, in the whole pathway, we are getting 2 ATPs here, and 2 here, and 2 NADH. Okay, this is the normal pathway of glycolysis. And I told you there are 3 steps in the whole glycolysis where the reaction is irreversible under the normal physiological conditions. 2 we have discussed before only. See here, one is hexokinase, the second one is phosphofructokinase 1, and the third one is here, this pyruvate kinase. This is also an irreversible reaction. So there are 3 reactions in this whole pathway which is irreversible under the normal conditions. Right. So now let's move further. What will happen with this pyruvate? This pyruvate, if there is presence of mitochondria, that pyruvate will move into the mitochondria, right? And of course, in the mitochondria, this pyruvate will go into Krebs cycle, and in the end, there will be ETC, and there must be the use of oxygen, right? That means in the presence of oxygen, or you can say in the presence of mitochondria, this pyruvate will change into acetyl-CoA. This pyruvate will move inside the mitochondria, and there it will change into acetyl-CoA. Acetyl-CoA. And in the mitochondria, acetyl-CoA will go in further reactions. The thing is that if there is presence of mitochondria, and with that there should be availability of oxygen, only in that condition pyruvate will move inside the mitochondria and will change into acetyl-CoA, and further reactions will occur. If there is presence of mitochondria and there is anoxic condition, that means there is no oxygen. Anaerobic condition, that means in that condition link reaction and etc can't happen in the absence of oxygen, right? So even in the presence of mitochondria, this pyruvate will change into lactate. What will happen with this? This pyruvate will change into lactate. If there is absence of mitochondria, there is no option, right? This pyruvate will change into lactate, as it happens in RBC, right? If there is presence of mitochondria and there is absence of oxygen, in that condition also pyruvate will change into lactate. And the name of the enzyme here is lactate dehydrogenase, right? And why this reaction is so important? Pyruvate will change into lactate. What happens is that this will use NADH and will turn into NAD+. Right? As we are getting, if you can see here, we are getting NADH, but we are using that NADH here and we are regenerating the NAD+, right? But why? We have got energy and we are using that energy and producing lactate. As you know, lactate is not very useful for us here. We are doing this reaction for some purpose. The purpose is that if there is presence of mitochondria or aerobic glycolysis is occurring, in that condition this NADH will move inside the mitochondria by shuttles. Directly it can't move, so through the shuttles this NADH will move inside the mitochondria, and there will be regeneration of NAD+, and so the glycolysis can occur one after other, right? But the thing is that if there is absence of mitochondria, right, and we are using all the NADH in glycolysis, so after some time there will absence of NAD+, so the glycolysis will stop. Glycolysis to occur, there should be the presence of NAD+. For that purpose we have to regenerate the NAD+. For that purpose this pyruvate will change into lactate and it will use NADH and it will regenerate NAD+. So as we will get NAD+, this NAD+ will again move to the glycolysis, and the glycolysis will occur one after the other, right? So this is the reason behind the formation of lactate. So let's move further, and I would like to discuss energetics of glycolysis, right? So let's see here and see energetics of glycolysis, right? We are talking about aerobic condition. We are getting 2 NADH. That means one NADH will give 2 ATP, 2.5 ATP. That means 2.5 multiplied by 2 is 5 ATP, right? 5 ATP. And ATP produced by substrate level phosphorylation. Substrate level phosphorylation, that means we are directly getting ATP, right? So by substrate level phosphorylation we are getting 4 ATPs. That is 4 ATP, right? And we are using ATP. How many? 2. So just minus 2 ATP. And what will the net gain here? 7 ATPs, right? So the net gain is 7 ATP here. Net gain is 7 ATP in the aerobic condition. So the net gain is 7 ATP, right? Net gain is 7 ATP. Let's see what happens in anaerobic condition, right? In anaerobic condition, as you have seen that glucose will change into pyruvate and pyruvate will change into lactate. And in that condition, in that reaction, NADH will get used, right? In anaerobic condition, NADH will get used in the conversion of pyruvate to lactate. So there is no NADH. So ATP produced is 4 ATP, and ATP used 2, so 2 ATP, right? So net gain will be 2 ATP. So the net gain is 2 ATP. So net gain is 2 ATP. In anaerobic condition, net gain is 2 ATP. 2 ATP. And in the aerobic condition, net gain is 7 ATP. Anaerobic condition, net gain is 2 ATP. Right? In the normal conditions, in RBC, we are getting 2 ATP, right? So let's move further. What is the end product? End product of glycolysis. End product of glycolysis. End product of glycolysis in aerobic condition. End product of glycolysis in aerobic condition is pyruvate. End product of glycolysis is pyruvate, right? And what is the end product of glycolysis in anaerobic? Anaerobic. Of course, lactate, right? And the next point is, what is the dead end of glycolysis? Dead end. Dead end of glycolysis. Dead end of glycolysis is lactate. Dead end of glycolysis is lactate. Okay, so discuss some more points, right? Where does lactic acid accumulation occurs, right? Lactic acid accumulation occurs in cancer cells. Lactic acid accumulation the first one is cancer cells. Cancer cells. The second one is exercising muscle. Exercising muscle, right? The third one is RBC, because anaerobic glycolysis will occur all the time, so lactic acid accumulation will occur with the RBC. There are some other parts of body also where lactic acid production is high, that is brain, GIT, renal medulla, skin, retina, etc. Right. One more point, and that is RBC completely reliant on glucose for their fuel, and the reason is because it lacks mitochondria. And one more thing about the brain, right? One more thing. Brain dependent on glucose as well. Dependent on glucose. Even in prolonged fasting, even in prolonged fasting, even in prolonged fasting, only 20% of its energy, only 20% of its energy from ketone body. Right? And what about heart? What about heart, right? What about heart? There is one special point about heart, is that heart is adapted for aerobic performance, right? So there is low glycolytic activity in the heart. So in the ischemic condition, there is poor survival of heart. That means I should write here, that means in the heart there is low glycolytic activity. Low glycolytic activity. So there is poor survival in ischemia. Right? So there is poor survival in ischemia. Right? These are the things. And further, I would like to discuss few points about the RBC. If you will see here, I have written a point, that is RL shunt. What is this RL shunt? Actually, in the RBC, as you know, the function of the RBC is to carry the oxygen, right? So in the glycolysis, whatever glucose RBC consumes, the 90% of glucose will go into the simpler glycolysis, and there will be production of 2 ATP in each pathway, right? But almost 10% of glucose will go through this RL shunt, and there will be production of 2,3-bisphosphoglycerate. Remember this name. 2,3-bisphosphoglycerate. This RL shunt will occur in RBC. This can also called as cycle, right? And what we will get by this 2,3-bisphosphoglycerate. The question is that what is the function of this 2,3-bisphosphoglycerate? Look here. This 2,3-bisphosphoglycerate has an important function. Actually, it will bind to beta chain of HbA and will help in release of oxygen, right? Enzyme here is mutase, and here this 2,3-bisphosphoglycerate will change into 3-phosphoglycerate, and enzyme here is phosphatase, right? And the phosphate will release in the inorganic form, and ADP will not be the acceptor here, so we are not getting ATP here. So if you will notice, in the RL shunt, net ATP production is 0. Why? Because we are getting 2 ATP in RBC. Net gain, there's 2 ATP, right? But as you can see here, we usually get 2 ATP here, and that 2 ATP will not form in the RL shunt, so net ATP gain is 0 in RL shunt. Net ATP gain is 0. Net ATP gain is 0 in RL shunt, right? This is the thing about, this is all about the RL shunt. And one more thing I would like to discuss with you is this. arsenic poisoning. Actually, what happens in arsenic poisoning? Actually, arsenic is inhibiting glyceraldehyde 3 phosphate dehydrogenase. I told you the enzyme here, Ga3P dehydrogenase. Right. So the arsenic will come here. What is this compound here? That is glyceraldehyde 3 phosphate. Glyceraldehyde 3 phosphate, right? And this glyceraldehyde phosphate was accepting phosphate, and usually it's changed into 1,3-bis phosphoglycerate, as you can see here, right? But what happens here? The arsenic will add. That means 1 arseno 3 phosphoglycerate. 1 arseno 3 phosphoglycerate. Actually, if you will see, this is the inhibitor of this glyceraldehyde 3 phosphate dehydrogenase. So NADH will not produce here. So NADH will not produce, and 1 arseno 3 phosphoglycerate will change into 3 phosphoglycerate. 1 arseno 3 phosphoglycerate will change into 3 phosphoglycerate, and arsenic will release by itself. So there is no ATP production here. There is no ATP production. So the net ATP gain will be 0, as NADH will not produce and ATP will also not produce here. So the net gain is 0. So one more thing I would like to discuss is that I told you that arsenic will inhibit, that means glyceraldehyde 3 phosphate dehydrogenase. One more thing, that is iodoacetate. Iodoacetate. Iodoacetate will also inhibit glyceraldehyde 3 phosphate dehydrogenase. Fluoride will inhibit this enolase, and this fluoride is used when we have to estimate the glucose, amount of glucose in blood. In that condition, we use fluoride there, right? And oxamate will inhibit this. Oxamate will inhibit this lactate dehydrogenase. Oxamate will inhibit this lactate dehydrogenase, right? So this is all about the glycolysis. You can see here, this is all about the glycolysis. Thank you very much for listening me patiently. I would request you all to please like the video, comment down in the comment box, please share the video, and subscribe my channel. And thank you very much. Bye bye.